So tell me what's going on in your lab.
Okay, so what we're doing is we're looking at interspecies transmission events for RNA viruses. And RNA viruses are very important because they have a high mutation rate and they frequently jump from animals to humans, causing things such as SARS or influenza or Ebola. And so we want to understand on a population level how this happens so we can start predicting if it's likely and if so what kind of virus it is.
Right.
So what we're looking at right now is a rabies virus outbreak that's happening in California, Humboldt County, where a skunk rabies virus has become established. It looks like in the fox population. And we have about 50 samples of skunks and fox brains, and we're going to start testing these samples to look at the viral populations to see if the virus jumped once or twice and how exactly it became established in the fox population.
So how does genomics fit in all that?
So what we're doing is using next-generation sequencing technology. We're doing Illumina right now, and we'll follow it up with 454. And we want to understand the depth of diversity in this population so we can make sure we divide our studies correctly.
Okay.
So we did some preliminary testing on a couple fox brains, got 450,000 X coverage.
Really? Okay.
Of almost the entire genome. And what we found was that there was some more common mutations, but most of them were less than 1% level. So you really do have to do deep sequence analysis. We also found that there were the mutations that occurred in one fox but not the other tended to be lower frequency than the mutations that were in both foxes, which suggests that the more persistent mutations might be something that's associated with the host gel.
Well, we thanks for talking to us, and thanks for coming by.
Thank you.